So today we are going to be talking about the stages of mitosis. My name is Christina Getby. A lot of you might have me for science, and if not, I'm happy that you were able to use this video for your reference. So let's go ahead and get started. So today's agenda will start with mitosis, approximately 15 minutes, and then we'll talk about questions and answers for about 15 minutes. And the question marks are going to be about what can I do to be a more successful student? And then finally, the last section of question and answers for about 15 minutes. So we'll take approximately an hour. So hopefully we can cover mitosis fairly well. All right, some things to remember. Please participate. You may answer questions by using your microphone, or you can send me a chat. Either way, I will see it or hear it. Please pay attention to your surroundings. If it gets noisy, please know that you can mute your microphone so it limits the distractions for everyone else that's joining us. And if you have to duck out of the live session a little early and are not able to see the whole thing, then they are going to be recorded on the American Academy YouTube channel. And then there is the link. So I have to tell you that last night when I was preparing for this live session, I had a video up from YouTube. And my children are 5 and 3, and right now they love YouTube. So both of them stopped at the computer and, Mom, what are you watching? What are you watching? What are you watching? And I said, oh, I'm just kind of learning, trying to refresh my memory. Well, what is it about? What is it about? And I'm thinking, okay, well, you're asking. So I turned to my 5-year-old and I said, it's about mitosis. And his response was, what's tosis? And I said, well, it's when your cells duplicate each other within your body. And he gave me a look that probably sums up how you all feel about mitosis. He curled his lip, turned away, and went to bed. So I share that with you because, like I said, I feel like a lot of people hear mitosis and automatically think, oh, that's awful. But it's really not. It's a very interesting topic. And within this video I was watching last night, the gentleman mentioned that if you want a reliable future job, become a biologist, because they are constantly gaining new knowledge about mitosis. It was the crash course on mitosis is what I was watching, and I really enjoyed those videos. But he also told me like he was just talking to me. He also mentioned that mitosis was coined and first kind of discovered back in the late 1800s. And then he said that there was a major breakthrough in 2012 that kind of told us about the spindles, the fibers that we'll mention in one of the stages. But there was a lot of time in between those two discoveries for lots of scientists to come and go and experiment with cells and mitosis. And still, after 150 years plus some, a graduate student discovered these spindles and their behavior. So it's always changing, and that's the cool thing about science, is there's always something else to learn. So please don't be afraid of mitosis. It's a fascinating process, and so hopefully I will be able to answer some of your questions by the end of this hour. So the learning objectives for this unit, we're going to explain how DNA is arranged in the nucleus, describe and recognize the stages of mitosis, describe the role of mitosis in the growth and maintenance of living organisms. Now, sounds like a bunch of words, but bear with me, we're going to break it apart, okay? So this is covering biology semester 1, and it's going to be talking about unit 3. Okay, and a lot of this information and the wording are taken straight from your lesson, unit 3. Okay, so you've probably heard a lot of this or seen a lot of this information. So just like humans go through stages, individual cells have different stages as well. So as humans, we start out as babies, and then we grow to adulthood, and then we grow and keep aging until we are elderly. So the life cycle of a cell can be divided into 2 stages or 2 parts. So we can have interphase and mitosis. So interphase is where the cell is going to spend the largest amount of its life in, okay? This is when the cell is going to carry out normal everyday cell functions, which again make up most of the cell's life. During this stage, however, the cell is going to prepare the DNA to be copied, okay, which it needs to be copied and prepared to be copied before mitosis even starts. So I have included a picture of what interphase would look like. So you see that round dark circle in the center, that's going to be our nucleus. Okay, so it's just resting. It's just living the cell life, I guess you could say. So before we get started talking about mitosis, let's go back and talk about DNA, review that a little bit. So every cell, as we saw in the last picture, has a central control center, and that's called the nucleus. This is important because it holds all of our genetic material, or DNA. DNA is a molecule that carries the information to make new cells and ultimately new organisms. But before that cell divides, all of its DNA must be copied so that it can be passed on to new cells. And it has to be copied exactly. Okay, when our bodies are going through mitosis and new cells are being developed, the DNA has to be the same, identical. You have a full set of DNA in every cell of your body except for your sex cells, which carry only half of your genetic information. Half comes from you and half comes from the other partner. All right, so chromatin. Now, things are going to look very different in the nucleus depending on what stage or what part of the cell cycle that the cell is in. This looser form of DNA, kind of lacks specific structure, is called the chromatin. And again, I've heard it referred to kind of like a hairball. Okay, it's all messy, it looks like there's no organization whatsoever. And again, even though we describe it as loose keratin, DNA is actually highly folded and looped to prevent it from being tangled and messy. Okay, so in this picture, I really enjoyed it because the chromatin looks like spaghetti or maybe chow mein. Okay, so some type of noodles that are all messed up and combined into one area. Now from there, the chromatin are going to form chromosomes, where they again are going to be tightly organized. Chromosomes. As mitosis starts, DNA condenses, and you can see specific rod-shaped structures, and these are called chromosomes. Each chromosome is made of a single long piece of DNA. So you can see in the top picture that that would be a chromosome, okay? And then in the bottom picture, you can see the double helix, the spiral-shaped DNA within the chromosome. Okay, chromatids and centromeres. Okay, so different organisms have different numbers of chromosomes. Okay, every human cell, again, except the sex cells, has a total of 46 chromosomes. Each chromatid, or just half of the chromosome, is an identical copy of the other chromatid. So a chromatid is going to be just one side of the X, okay? And then they are joined together at the centromere. Okay? This is very important because we'll be learning or using these terms when we start talking about mitosis in the different stages. So again, it's important to remember that because the DNA was already copied, each chromatid contains a full set of genetic information. So one side of that X is going to contain a full set of genetic information. All right, so here we have a diagram kind of showing you the vocabulary words. So you can see that the 1X right here is going to be, let me get the pointer. Okay, this right here, this whole X shape is the chromosome, that whole thing, okay? Now, one side of it is the chromatid, and they are going to be joined in the center or the centromere, okay? So chromosome, again, is the whole X shape, and the chromatid is just the one side. And our last slide told us that the one side, the chromatid, is going to hold a copy of your DNA. All right, so here we go, mitosis. Again, breathe, it'll be okay. So mitosis, this is when the cell division actually happens, okay? Cell growth stops and one cell divides and becomes two cells. Now, mitosis itself consists of 4 stages represented by PMAT, prophase, metaphase, anaphase, and telophase. So in this picture, again, it was taken from the lesson. In this picture, the first image represents a cell in interphase. So the first picture of this series is going to be the resting phase, okay? Prophase. So prophase is the first phase of mitosis. This is where the chromosomes are going to condense and become visible. We're actually going to, from the interphase, from that bowl of spaghetti, we're going to have some more structure where the chromosomes are going to be able to be seen. So again, at this point, the DNA has already been copied. So important steps that happen at this stage, we have the nuclear membrane breaks down. Chromosomes are going to become visible. And then we have spindles that are going to form and they're going to move to each pole of the cell, okay? So I've included kind of a drawing, more like a cartoon drawing of prophase, but you can see the chromosomes, you can see the spindles, and you can see that the membrane's kind of breaking down, kind of disintegrating. So let's see if this kind of helps make sense of prophase.
Welcome to the In this video, I'd like to talk about the first stage of mitosis called prophase. Let's first take a look and see what it looks like. Several important things occur during prophase. First, the DNA continues to condense and form chromosomes. Second, the nuclear membrane begins to break down, and eventually it will go away for a short period. Next, centrioles begin to create spindle fibers. These fibers will be important because they will attach to the chromosomes at the centromere and will begin a tug of war and move these around the cell. Let's take another look at prophase. So in summary, the DNA condenses into chromosomes, the nuclear membrane breaks down, and the centrioles begin to produce spindle fibers. Thanks for watching. The Moo Math and Science uploads a new math and science video.
Okay, so again, hopefully that made sense. So from prophase, oops. From prophase, which is the first technical stage of mitosis, we have metaphase. Now, metaphase, again, is the second phase of mitosis in which the chromosomes are going to line up at the center of the cell. Okay? Once the chromosomes are lined up in the middle, the fibers are going to reach out and attach themselves on one side of each centromere. Okay, let's see. Hopefully this will make sense. Oops, I'm so sorry.
Welcome to Moo Math and Science. In this video, I'd like to talk about metaphase, the second stage of mitosis. Here's what metaphase looks like. What occurs during this second stage of mitosis? First, the chromosomes line up in the middle of the cell. They align halfway between the two poles of the cell. How does this happen? Long microtubular fibers attach to the chromosomes at the centromeres at a specialized region called the connector cores. The fibers extend from the poles of the cell and play a tug of war that align the chromosomes in the middle of the cell. Here are some additional pictures of metaphase. So in summary, metaphase is when the chromosomes of the cell line in the middle of the cell and spindle fibers attach to them and get ready for the next stage, which is called anaphase. Thanks for watching. The Moo Math uploads a new math and
Okay, so again, in metaphase, we're going to see where all the chromosomes are going to line up in the center of the cell. Okay, third stage or third phase is the anaphase, where chromosomes begin to be pulled apart and pulled toward the opposite poles. So here is where the grad student at MIT noticed that these spindle fibers were performing this behavior of kind of pulling apart those chromosomes. So here you can see that each chromosome is split up in half, and a chromatid is going to go to each side of the cell. Okay, so again, all the fibers will carry a chromatid to each pole of the cell. So this is why it's important that the chromatids are carrying an exact copy of our DNA.
Welcome to Moo Math and Science. In this video, I'd like to talk about the third stage of mitosis called anaphase. Let's take a look at what anaphase looks like. What occurs during this stage of mitosis? First, the sister chromatids, which are aligned at the equator of the cell, begin to pull apart. These sister chromatids are created during interphase. What happens is that a single chrome is duplicated into two copies and then are joined together by a centromere. Next, the chromosomes are attached to spindle fibers at the centromeres, more specifically at the connector core. And during anaphase, they begin to pull towards the poles and separate the sister chromatids at the centromeres. Here's what this looks like. So in summary, anaphase is when the sister chromatids break apart and begin to pull towards the poles. Thanks for watching, and Moo Math and Science uploads a new math.
All right. Okay, and then the fourth phase is the telophase. And this is where the chromosomes are pulled to opposite poles, and then our new nuclei will form. So here we have the chromosomes are going to disintegrate, and then they're going to go back to that bowl of spaghetti, that jumbled and organized mess. We have the nuclear membranes are going to form around the DNA. And then we have the cell elongating. So I've got one more video from Moo Moo Math.
Welcome to Moo Moo Math and Science. In this video, I'd like to talk about the fourth stage of mitosis, which is called telophase. Let's first take a look and see what telophase looks like. A couple important things begin to occur during telophase. First, the chromosomes that first began to appear during prophase now begin to retract and dissolve. Secondly, the nuclear membrane that was dissolving during prophase now begins to reform, except this time you have two nuclear membranes on each pole of the cell. Next, if you take a look at the shape of the cell during telophase, you will notice that the cell is beginning to elongate and that it is pinching in the middle. This is because two new cells are forming, and this will be completed during cytokinesis. So as a recap, during telophase, chromosomes dissolve, the cell begins to pinch in at the middle, and the nuclear membranes begin to appear. Thanks for watching, and Moo Moo Math uploads a new map.
All right. So we've covered all the stages or phases of mitosis, and now we have cytokinesis. And so after the new nuclei have formed in telophase, we have this process called cytokinesis, where the cell physically divides into two. So now we have two new daughter cells that have a full set of DNA, and they each begin then their life, the majority of their life within interphase. So here you can see in this picture, you can see the gobbled, messy DNA that's in the middle of the nucleus now. It's not the X shapes that we were seeing. It's more like, again, that bowl of spaghetti all jumbled in there together. So we've got the two new daughter phases, or two new daughter cells that contain the full DNA. Okay, you're probably thinking, all right, this is a lot of information. Who really cares? So why is this process important? Well, there are single-celled organisms out there that use this process to make a whole new organism. But for us as humans, and obviously we're multi-celled organisms, we need new cells for various reasons. If we injure ourselves or scratch ourselves, we need new cells to grow and develop to heal ourselves. When we were born and we were an 8-pound baby, our cells don't just get bigger so we are bigger people. No, our cells are dividing and multiplying, making us grow. Okay, so there's a lot of different reasons. We need to maintain ourselves if we need to repair our body. So again, so you started out as one cell, and this process happened many, many, many times over your life. And as your body continues to grow and maintain itself, it continues to grow new cells. So that's why it's important. I know that, again, there's a lot of information. Maybe it's a hard process to contemplate, but it is very important. So this is the film, that is a short video that was very similar to the one that was provided in your lesson. So let's watch it again and then we can talk about it.
Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes. Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates. With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug of war, the spindles pull each chromatid toward opposite poles. Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes then unwind and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating two new cells in this ongoing cycle that sustains all life.
So, as we were talking about job security, you could become a cellular biologist and discover why the nuclear membrane disintegrates as a portion of mitosis. Okay. All right, so I wanted to go over the assignment 3.2.1, the stages of mitosis, because a lot of us look at this and think, oh, I don't even understand the directions, so I'm not even going to try. It's a lot easier than what the directions implement or imply, I guess is the better word. So, okay. We are going to be looking at an actual cell under a microscope, and we're going to look at the image and see cells in various phases of mitosis, okay? So, hints that have been provided for you. Each individual cell is surrounded by a rectangular cell wall. The nucleus is the darker stained area of each cell, and cells in interphase do not have distinct structures in the nucleus. Okay, fair enough. Your task is to estimate the number of cells you think belong to each phase of the cell cycle and record your data. Explain how you decided it, what occurs at each phase of mitosis. Okay, so our first thing is to count. Now, when you look at this cell, it is a fuzzy, very grainy picture, but the images of the different phases will still jump out at you, okay? Another example would be, okay, here we can see the different cells in the different stages of mitosis, okay? So we have right here, we have the DNA in a gobbled mess. That's going to be our prophase. We have the DNA lining up right in the middle. That's pretty easy to see. We have the chromatids being pulled to to the opposite poles. Okay, again, that's pretty easy to see. So here we have the cytokinesis occurring where the two different cells are actually forming. Okay, so these are different examples of the one that you saw or have in your homework. So another example would be this one. Okay, again, you can see the different phases and they're there. You just have to really use your best judgment. If they're not as clear cut as this one where the chromatids are at opposite ends, or this one where there's two nuclear membranes, okay, they might not be that clear, okay, but it's definitely worth a shot. All right, so let's go back to this one. All right, so we again have the different stages here. So interphase, again, is the resting stage, which me personally, I would count all the actual stages of mitosis and then count what I have left to see cells. And the point of this activity is not to count them exactly, but to start kind of training your eye to know the differences that the cell will visually look like, okay? So let's say anaphase. So anaphase is going to be where they begin to be pulled apart and they are going to go to opposite ends of the opposite poles. So here, that looks like a definite anaphase cell, okay? Again, there's kind of like a double image going on. So maybe we could say that this one is one as well. Maybe this one and maybe that one, okay? So then we have, I guess I should have started at the first stage, but I didn't. Okay, so prophase, stage one. Prophase, again, is where we can see the DNA, okay? And we are going to see the chromosomes. So the chromosomes, I would say maybe this one. Again, lots of blurry images, but maybe this and maybe this one, okay, just to give you an idea. Again, it's not about counting the cells exactly right, but it's just looking for those differences so you fully understand the different phases. So then metaphase is where they are going to be lined up in the middle. So metaphase is a little bit easier to see. Maybe these guys, okay, so lining up right in the middle. Anaphase, we already talked about. And then we have telophase, where we have new nuclei are going to start forming. So we're going to look for maybe this guy, maybe this guy, maybe some over here, okay, where within one cell wall, we have those two dark spots. What I look at when I'm grading this assignment is the description, your explanation on why you counted that cell for which stage. Okay, so prophase. How can you describe, what can you say about prophase? How did you know which cells fit in prophase? Okay, well, I'm going to go back to my lesson and I'm going to go and I'm going to say, okay, well, at prophase, I know that the chromosomes are going to condense and become visible. I've got my nuclear membrane that's disappearing, my chromosomes that are becoming visible, and my spindles. Okay, so for your explanation, you could say something like, I knew there were this amount of cells within the prophase stage of mitosis because I was able to see the chromosomes and I maybe saw some spindles starting to form. Okay, that's it. Right, so the next one we have is going to be metaphase. Okay, if I look at my onion cell, okay, again, we kind of already talked about which cells are going to be in the metaphase of mitosis, but how did you know? So my explanation could be something about, I knew that there were this amount of cells within the metaphase stage because all of the chromosomes were lined up right in the middle, okay? Next, we have anaphase, okay? Anaphase, find out how many cells are in your onion cell, and then come back and say, I know that there are this amount of cells within the stage of anaphase because I can see that the chromosomes are beginning to be pulled apart to opposite poles. Okay, this is where those fibers pull, kind of like the tug of war. Okay, then finally, telophase. This is where I'm going to see the chromosomes disintegrate. So they go back to that crumbled mess, hairball-like substance. And then I get to new nuclear membranes. So I know that there are this amount of cells within the telophase stage because I can see two nuclear membranes and I don't see the chromosomes anymore. Okay. And then I believe it asks you to talk about interphase. Well, how would you describe interphase? I know that this is interphase because I just see a dark circle of nucleus. Okay. It's just chilling, just living the cell life. Okay. So that's it. All right. When you do this, again, the most important part of this is your explanation. Your explanation tells me that you are fully understanding the different stages of mitosis. Okay. So you can always maybe Google some onion cells and kind of get your eye trained to see those different cell stages and then go back to the one on your homework if that helps. Okay. I also wanted to look at 3.2.1 DNA, life cycles, and mitosis. So we'll go back to that assignment. All right, so we have, if an animal cell with 4 chromosomes in its nucleus goes through mitosis, how many chromosomes does each daughter cell have? 2, 8, 4, or 16? What do you think? If our daughter cells are supposed to be exact copies of our cells, and this animal cell has 4 chromosomes, then each daughter cell is going to have 4 chromosomes. So the answer would be 4. All right, what phase of mitosis is highlighted in the slide below? Is that going to be metaphase, telophase, prophase, or anaphase? What do we think? I believe since it looks like all the chromosomes are lining up in the middle, I would call that metaphase. Number 3, the majority of the cell's life is in this phase: cytokinesis, centromere, prophase, or interphase. Which one? Well, let's say you're not exactly sure about this one. So we can do process of elimination. Cytokinesis, we learned that that is the process of the cells actually breaking apart, forming 2 new cells. So I'm pretty sure that's not it. Centromere, it's not a phase of mitosis, so we can rule that one out. Prophase is the first stage. And interphase is going to be where the cell spends most of its life, performing those daily functions, copying the DNA within that interphase. Okay. All right, now number 4, the phases of mitosis in order. So the 4 phases of mitosis, we're going to start with prophase, then we are going to jump to metaphase, then anaphase, and then telophase. So you can remember as PMAT, prophase, metaphase, anaphase, and telophase. Mitosis occurs in the blank of the cell, the nucleus, the brain, the cell wall, or the cell membrane. Which one do we think? Again, we can kind of do process of elimination. Okay, I know it's not a cell wall because animal cells don't have cell walls, so I can scratch that out. I know it's a brain because cells don't have brains. They have a central control station, if you will, not a brain. So we either have a cell membrane or a nucleus. Well, I believe the cell membrane is what kind of holds the cell together. So I would think it would be the nucleus. Okay, so the nucleus is going to be where mitosis occurs. The nucleus, again, holds all of our genetic DNA material. All right, number 6, it is critical that all of the cells' DNA is copied during the synthesis phase prior to cell division because the cell is going to divide into 2 new cells. So the DNA must first be copied so each daughter cell receives a full set of genetic information. Would that be true or false? That is definitely going to be true. We really want each daughter cell to have the exact set, an exact copy of genetic information. All right, so if you look through a microscope but cannot see any distinct chromosomes in the nucleus, what stage are you viewing? Prophase, interphase, metaphase, or anaphase? I don't see any chromosomes when I look in the nucleus. If I don't see any chromosomes, it's going to be the cell's resting phase, which is interphase. All right, number 8, DNA is copied during blank, which happens prior to cell division. Metaphase, division, growth, interphase. Which one? Again, it's going to be interphase, that resting phase of the cell. When we look at all of this copying of genetic information and all of the intricate parts that make up mitosis, it's very easy to see how genetic mutations occur, or a extra chromosome gets put in, or all of these occurrences. You can see how easily they can occur, for sure. So it's pretty fascinating. Science is amazing. I hope that you guys will see that and get excited about it like I do. So moving on, do you have any questions? If you have any questions, please feel free to email me or send me a text, even if it doesn't occur or relate to mitosis. I'd love to hear from you so I can help you be successful. Which leads me to my next portion of the presentation. All right, so I got on the website or the internet and I found this website called Opportunity International, and they had 10 habits of successful students. And you can find all kinds of lists of healthy habits or, healthy tips or studying tips. They're all going to be different amounts. Some of them are going to be 7, some are going to be 12, but they're all going to be the same general idea. Okay, so the first step or first habit, I guess, according to Opportunity International, is going to be get organized, okay? Making a plan for what you're going to do and when you're going to do it will make sure that you're always ahead of the curve, okay? It's very important to be a good time manager, if that makes sense. So making a plan for what you're going to do, when you're going to do it, helps you manage your time a lot better. Okay, the second habit, don't multitask. Studies have shown that multitasking is physically impossible. Now, physically, I think you can multitask. However, mentally, it's hard to comprehend everything that you are multitasking. I was on the phone talking to my mother last night, and I was texting my sister-in-law while I was on the phone with my mom, and I was starting to cook dinner, and I found myself getting kind of lost with what my mom was saying. So yes, it is very difficult to multitask. So if you're studying for your mitosis portion of your exam, study mitosis. Don't go and write your paper for English and draw your art project while you're studying for mitosis. Just focus on one thing at a time. All right, the third habit, divide it up. So studying isn't fun to begin with, and forcing yourself through a study marathon will only make it worse. Dividing your work into manageable chunks and rewarding yourself when you finish each chunk will make studying more fun. So yes, it doesn't mean that you have to spend 3 solid hours studying. That's a lot of solid studying time. Divide it up. Study for a half hour, then go walk around the house for 10 minutes. Or study for a half hour, go switch out the laundry, fold the clothes, do some dishes, okay? I know those aren't fun things either, but they will help you break up the time so you're not just sitting there studying. 4 is sleep. Do not underestimate the importance of those 8 hours of Z's every night. Getting a good night's rest will sharpen your focus and improve your working memory. Never miss out on sleep. It's not a cool feeling. Number 5, set a schedule. Do you work better right after school or after you've eaten dinner? Are you more productive in 90-minute blocks or half-hour spurts? Find a schedule that works for you and stick to it. Okay, my oldest son is in kindergarten, and we've kind of figured out that when he gets home from school, he needs a break. He needs a snack. He needs about 15 minutes to be a kid, and then we can come back and focus on the homework that he needs to finish. So set a schedule for sure, that helps. Take notes. Taking notes will not only keep you more engaged during class, but will also help you narrow down what you need to study when exam time rolls around. It's much easier to reread your notes than to reread your entire textbook. And I fully agree with this one. If you are super tired because you stayed up and you skipped your night of sleep, you stayed up all night studying, if you force yourself to be engaged by taking notes, it will help you focus more on class. When I used to teach at a brick and mortar school, I would have students all the time, oh, we still have an hour left. Well, yeah, we do, but are you participating in the class? Because if you're participating, taking notes, the time is going to go by a lot quicker than if you're just sitting there sulking because you're tired. So taking notes is one of those ways to become engaged and participate in class. Study. Okay, Opportunity International says that this one might be obvious, but did you know that there's a right and wrong way to study? Review your material several days ahead of time in small chunks and in different manners. For example, write flashcards one day, take practice cards or practice texts the next. In other words, don't cram. In other words, don't reread the whole chapter the night before the test. Or don't reread all the notes that you took the night before the test. If you take a page of notes during a day in school, then go home, review that page of notes, okay? So it breaks it up and forcing you to learn the material. Number 8, manage your study space. Find a place that will maximize your productivity with places away from the television and other distractions. Whether it's your local library, just a desk in your bedroom, set aside a study space that you'll want to spend time in. Okay, you need to be comfortable in this space. You need to be away from distractions, and you need to feel at ease so your mind can comprehend what you are trying to study. Find a study group. Sitting down with a group of people who are learning the same things as you is a great way to go over confusing class material or prepare for a big test. You can quiz each other, reteach material, and make sure that everyone is on the same page. After all, teaching someone else is the best way to learn. Okay? For whatever reason, again, when I had a classroom, I would explain something the students wouldn't understand. So I would ask someone else in the class to explain the same concept. They would use the exact same words that I just did, but for whatever reason, it made more sense to their classmates when they said it. So if you're going to a study group asking what this term means versus what this term means, they're going to be able to use a language that's more familiar to you, okay? And number 10, ask questions. This is one of my favorites. You're in school to learn, so don't be afraid to do just that. Asking for help from a teacher, a tutor, or your friends is a surefire way to make sure you truly understand the material. If you don't understand an assignment, ask. If you don't understand the instructions on activity 3.2.1, Stages of Mitosis, ask. Send out an email, send a chat, send something so that I know that you need further instructions, okay? That's my job. Again, I want to make sure that you can successfully pass all of your science classes, okay? So again, those are the top 10 habits of successful learners according to the opportunityinternational.com. All right, any final questions? Well, I appreciate you and thank you for all that you do. If you have any questions or concerns or comments, feel free to email me. I'd love to hear them. I'd love to help you with your classwork. And again, I'm here for you. Have a great day.